Well, thank you very much, everyone, for joining us today. And thank you, Assaf, for accepting our invitation. I will do a brief introduction to Assaf. Assaf was born in Haifa. He's a professor in plant and environmental science at the Weizmann Institute. I had the privilege to meet him in person during one of my first mob meetings in Eilat in 2016. Assaf earned his bachelor degree and PhD in 2004 at the Hebrew University of Jerusalem and completed postdocs in Paris and Rutgers University, then joined Weizmann Institute in 2010. His research centers on marine microbial ecology, especially phytoplankton, virus interactions, and their influence on oceanic biogeochemical cycles and climates. Assaf collaborated with his wife, artist Nivi Alroy, in an amazing Weizmann talk that I will leave a link about in the chat, which is about art and science and microorganism. And it's called Through the Looking Glass, Life in a Drop of Water. That was an old talk, but I think still amazing talk. I think they also collaborated on a children's book about marine microorganisms and probably more. So Assaf, please, you can start and let me know if I missed anything.
Thank you very much, the organizing committee, Fuad, thank you. The book is still unpublished, but we have it in like 5 languages ready from the old times in Paris. I'm excited to talk to this specific crowd of microscale because my talk will be all over connecting scales all the way. Can you see my cursor or no? Can you see my cursor?
We do.
Okay. Also when it's here. You see it or no?
Yes, we do see it. It's small, but we do see it.
Yeah, so my talk, the main theme of our work and the leitmotif in our work is really to connect scales all the way from the molecules to the single cell organism up to the actual algal blooms that we studied for many years in the ocean. And throughout the talk and the work, we will do zoom in and zoom out by looking at host-virus interaction across the scales. And move the slides. Okay, can you see it now? Yeah. So for years, we studied the outstanding phenomena of algal blooms in the ocean that can stretch over thousands of square kilometers. This is the foundation of life and all interactions in the ocean. Hugely important, of course, because in algal bloom, annually, you can fix more than 50 billion ton carbon a year. So it's the foundation of marine food web. And one of the key questions we ask is during phytoplankton bloom or algal blooms, what will be the main mortality agent that control cell fate in the bloom? Because that will have a tremendous consequence on the flux of organic carbon and nitrogen and phosphate that can really fuel the entire ocean microbiome. So what is the metabolic valve that control this key ecosystem process?
For a seminar.
Sorry?
Could you please turn your mic off? Thank you.
Okay, so yeah, and we go all the way, as you will see, to the molecular mechanism. The main goal is to understand the molecular mechanism or the cellular mechanism that regulates cell fate in the bloom. But there are many ways to die in algal blooms, and can be different type of interaction. And in general, because of the high density of algae in these massive blooms in the ocean, this is a great hotspot for interactions. It can be intraspecies interaction between algae. It can be top-down regulation by grazers. It can be, and we'll talk a lot about algae virus interaction, but also parasite as fungi and interaction with symbiotic or pathogenic bacteria. And one of our leading working hypotheses is that each of these interactions will leave unique metabolic landscape that will be unique to how interaction occurs and which of the metabolic resources are needed to fuel the interaction. So we are very much interested, end of the day, to be able to link scale all the way from the micro scale to the macro scale, and to better understand how it will control the cell fate and the flux of organic carbon and inorganic carbon within marine microbial food web. So just in one slide about the general idea how we approach such big questions, ecological question, with more mechanistical point of view. So we will do throughout the talk ocean to lab processes and backwards in order to try to untangle the different processes that we just saw and how the single cell interact with viruses, bacteria, how they communicate within the same population. And so we start with going and chasing these algal blooms in the ocean. We bring back isolates to the lab, and we're generating, and that's key in our work, a tractable model system in the lab that will mimic some of the key interactions in the field. Doing so by different omics approach, as I will show you, we also dive into understand the uniqueness of each of these interaction and what mediated and what is the unique metabolic landscape that is produced in each of this interaction, because that can give us a really powerful tool to develop metabolic biomarkers that then we go back to the field and we can try to detect interaction in action. So we'll start now to focus specifically on interaction with viruses and the importance of viruses in the marine environment. One of the key ecosystem pathway that is regulated and driven by viral infection is the well-known mechanism or ecosystem mechanism that it's called the viral shunt. This is really a key fundamental process in which the huge amount of carbon that is fixed in algal blooms in a very, very simplified manner, instead of going up in the higher trophic level, is being shunting by lysis of viruses to produce massive amount of viral-derived dissolved organic matter and also particulate. And that will fuel the microbial life, as we will see later in the talk. But this ecosystem pathway, the viral shunt, was proposed by Steve Willem and Curtis Sattel more than 25 years ago, but it still remained almost enigmatic and unquantifiable. And today I will show you how, with implementing key single-cell approaches, we can start to really quantify this process in order to understand the impact of viruses on the metabolic landscape and also on the microbiome composition and structure, in order to eventually learn more about where the carbon will go. So there are many ways to detect viruses in the ocean. And what we try to do, as also other labs, is to move from the static point of view of viruses as just like passive virions, and to move into understand the virus cell. And we'll give you some example. There are several approaches to learn about interaction with viruses, and many of them deal with metabolic fluxes, single-cell polonies, single-cell RNA-seq, as I will show you today, metabolomics, and so on. And one of the ideas is to move, if you take a drop of water, and you all know these images from Foreman et al., if you take a drop of water from anywhere, from any ecosystem, and you simply stain it with cyber green, you will see all these beautiful dots that are presumably viral particles. The big guys are bacteria, and these ones look like a diatom. But in our work, we try to move from this passive point of view into understand how the virus cell, the infected cells, is almost a new entity that is being formed. And this is the active infection and all the metabolic steps that is needed for the replication of the virus, because that's the real impact on the environment. So we try to develop tools to quantify the infection in all states in the environment, to identify the unique metabolites produced during infection and how it reshaped during infection. And also recently, we start to learn about defense system, antiviral defense system. So the virus cell, very important concept that we'll focus on today. And the model system, the very attractive model system, hugely ecologically important model system that we study, host-virus interaction, is between Emiliania Huxley, this single-cell eukaryote algae, coccolithophores, and their giant viruses. Emiliania Huxley, or HEAX in short, can form these massive blooms in the North Atlantic, as we can see here from satellite. It's not only hugely important for photosynthesis, it's also decorated by a beautiful calcium carbonate shell. And therefore, the interaction between the viruses that control bloom demise is hugely important. As you can see here, one of the blooms we visited a few years ago, we see that within a few days, the bloom that we see from satellite completely shrink and demise within a few days due to viral infection. This giant virus that infect is very specific to Emiliania Huxley. It's a giant virus, contains more than 700 genes, and it's encoding for unique metabolic pathway never seen before in marine viruses, for almost a complete biosynthetic pathway for sphingolipids that eukaryotes will have, but never encoded by viruses, except for this virus. And of course, we are interested to study because that will have very important consequence, not only on the carbon that is going down to the sediment in the ocean, but also in production of DMS, the smell of the sea, this organic sulfur that this cell will produce, with the potential as a secondary aerosol to control cloud condensation nuclei. So really want to understand how the single cell, the fate of the single cell during infection will affect other member of the cascade in the trophic level, but also ocean atmosphere feedback. Here, the famous Cliff of Dover made by calcite by these blooms. In a way, it's a relic of bloom demise, if you wish, over geological timescale. So let's start slowly, zoom into all the way from the bloom, from the thousands of square kilometers of bloom in the ocean. How do we slowly zoom into a single cell resolution? And what we developed in recent years is to understand individuality within a population using various tools, as I will show you. And in order to understand and disentangle the virus cell sphere to its component. This is, as we say, the virus cell, the infected cells. It can be in different stages of infection or different kinetic class, early and late and mid. Some of the cell will die in a programmed cell death manner, but also small subpopulation will always survive the infection in our hand. And we see evolution or involvement in production of resistant cell, but also some chronic cells. And all the interesting metabolites that are produced during this interaction. If we go with the traditional bulk analysis measurements of RNA-seq or proteomics or genomics, we will miss all these subtle phenotypes or different outcome of infection that we really want to start to quantify. So we want to move from bulk analysis, where we average all this interesting phenotype, to more single cell approaches to understand individuality during host virus interaction. And in, sorry. And in order to do so, in the last few years, maybe 4 or 5 years, we developed several very interesting approach that is new to the field of marine microbiology, and different ways, single-cell transcriptomic approaches for several reasons. And in order to get, a single-cell view on host virus dynamic in the ocean. And starting with understanding even the most basic question of what is the fraction in a water body with population infected, even to answer the most basic question of what is the fraction of infected cells, sometimes you need to apply the most sophisticated cutting-edge approach, as you will see. So we start to understand the fraction of active viral infection, again, not just to count viral particles, but to count the virocell in the ocean. It also gives us a powerful tool to understand phenotypic heterogeneity, so different options to the infection, and also to map directly between viral infection to the host response on a single cell resolution. Again, if we do it in bulk, we will never be able to do this match. And lastly, I will give you some nice example from recent work from the lab in which we use the fact that we can look at two microbes interacting within the same cell, a virus and the host, the infecting, in a single cell resolution in order to discover new and novel host virus systems, pairs in the marine environment. And we do it by several approaches that we develop. Some of them are based on single cell sorting, and some is more based on droplet-based automatic microfluidics. The flow cytometry will give us another dimension because then we can pick and select and sort specific subpopulation, either using also different fluorescent dye for physiological state. And the droplet-based is more high throughput, so we can get up to 10 information on transcriptomic information per sample, up to 10,000 cells even. And also, I will show you other approach with single cell molecule fish that also we can co-probe the host and the virus. So just a few examples how the data look like with this first paper we published 4 years ago. It's just to give you a glimpse on also when you start to look at completely different resolution, you need to learn the data over a long time because there are no textbook to understand what you see. And this is just one simple example that I really like, in which we simply look here, each dot is a single cell from a single cell transcriptomic, and this is 4 hours post-infection. Each dot is a single cell, and the y-axis is the level of viral transcriptome within each cell. And already here, this is logarithmic scale, we can see beautifully that even that the population of EX is identical genetically, you infect them, and already here you see a high phenotypic variability within the infected cells. Some cells are very infected, and some are not infected at all, at least at this stage of the infection. If I would do RNA-seq in bulk, the usual way, this is what I will get. 1 point here, that average, so I miss all this interesting phenotypic variability during the infection. And I'll show you how we use it. So now, we reorder bioinformatically, we reorder this single cell. Sorry?
Just continue, yeah.
And so now we reorder the transcription. Again, each dot is a single cell and its transcriptional profile, but we reorder them based on their expression profile. So we start to generate this gradient of infection state, what we call the continuum of infection state. So we are not only in a state of infected or not, but also all the intermediate, and we call this neighborhoods of cells, this cluster of cells, we call them meta cells. So they are shared the same profile of expression. and that enable us now to define the infection state of the cell in a high-resolution manner. What we did next, and came by surprise, that this is the same data, but now every column is a single cell, and every row is different viral genes. And when we reorder them again, based on the infection state, we start to see this beautiful cascade of transcriptional program that is being activated and turned on and off. And that enable us, when we start to look at the promoter of each of these cluster of genes, we identify and define different kinetic classes of infection, starting with the immediate, early, and late. And so how something we would never be able to do in bulk analysis, but when we reorder them based on the single cell transcriptomic, we can start to define their kinetic class. That was in a lab culture. And what Gur Hevroni in the lab did, so now that we have definition of not just infected cells and how many of the infected cells, but also at which kinetic class they are, we took it, and that was most challenging, to the environment. Here we induce in Bergen and EAX blooms in the fjords of Bergen. And what you see here, each dot again is a cell, and this is a level of transcriptome of the viral genes. And what we start to see here is really the pandemic of the infection for the first time during the succession of the bloom at each state, the level of the infection. And also we can start to break it to which of the state of the infection we are. And we got very interesting surprises, like, for example, that we start to really detect the infection ultra sensitive, not when the bloom demise, and that's the traditional way to think about this almost predator-prey interaction and demise and increase in viruses, but as early as the early state of the bloom succession even. And when you start to look at the fraction of each kinetic class in each of the days, surprisingly, we saw that they are very similar, the fraction of different kinetic class. And for us, it gave us an interesting estimation that actually we look at the complete replication cycle of the virus, a daily turnover of the infection based on single-cell transcriptomic data. I heard that also Flora, the former great postdoc in the lab, talked to you a few weeks ago. And again, Flora, when she was a postdoc with me, developed another approach to look at viral infection, which is single molecule FISH, in which we probe both, we had a probe specific to the viral genes and also to photosynthetic genes in the host. And that, again, gives us a powerful tool to look at co-infection and the regulation of the viral at different state of the infection. Here we see nice shining cells at the advanced viral infection state, and a shot of the host genes. And we can do it in microscopy, but also we can take it to flow cytometry. Now it's running routinely in the lab. And also, this is the data from imaging flow cytometry, enable us to do it in high throughput and start to look at this interesting phenotypic variability between the viral genes and the host genes in each fraction here over the time course of infection. So again, simply by now, we don't need the whole transcriptome. We're probing a viral gene and a host gene. We co-express within single cell, and we quantify the fraction of them in the population during the course of infection. And again, also, we take it to the field as a powerful tool to visualize the beauty here that also in microscopy, you can visualize and learn about the replication cycle, the life cycle of these giant viruses. And, and we also took it to quantify infection within the bloom. Sorry. And now, this is, the classical bloom demise, almost in a synchronized manner, and extracellular production of viruses, like a clear textbook information on hosts and virus. And now you will see the great surprise. You see here the bloom from Emiliano Huxley. The color becomes milky from the calcite. Very extensive bloom that we induce in Bergen in a mesocosm setup. And now came the big surprise when we apply the single cell and single molecule fish. When we look at the fraction of infected cells, so again, the fraction of the virocell, we get only up to 25%, although the entire bloom crushed almost in a synchronized manner. So what is going on? And this is for us was amazing and opened completely new field in the lab to understand, because again, the power of having tools to quantify the mortality agent, in this case is a virus, can really help us now to disentangle the different mortality agent that acting in concert maybe during bloom demise. And that opened interesting questions in the lab to what's going on here. Again, if you stay with just extracellular point of view, you will never guess that viruses are not acting alone here, because you will see viral production and bloom demise. But only when you start to look intracellular at viral infection in action, then you add a completely new dimension to the interaction. Another, you can call it application of understanding the real fraction of infected cells for us, is to be able to couple it to the carbon budget. And that's what we did here in the same experiment. And what we found very interestingly, because now we know the fraction of infected cells, and with the simple modeling, we calculate that actually the infected cells produce almost five times more calcite or PIC, and 5 times more TEP. TEP are the extracellular polymer or sugars. And as you can see here, this is from an old work I did during my postdoc, in which you can see that the single cell of EX during infection are actually trapped in aggregates. And these two, both the calcite and the sugars that make them sticky and make aggregate, can really change now the potential for carbon export to the depth. So we're playing here with probably one of the key fundamental important process in understanding this process in microbial food web, the viral shunt, the recycling, versus the viral shuttle, which is the drawdown of these aggregates in the carbon export. So what is really controlling the shift between the two is really key question that we can start to ask now. Back to our complex virocell sphere, let's look now on this part of the different component. And also, as I mentioned, these cells, as we will see soon, are producing also unique metabolites that we want to map and understand, but also extracellular vesicle. And Daniela Schatz, with the amazing work in the lab, really opened a new field in marine microbiology with identifying that the infected cells are not only producing virions, but they're also producing a very unique cargo encapsulate in these lipid vesicles, extracellular vesicles. For years, we thought in the electron microscopy that we're looking at just garbage out of the cells. It took us a while to understand that this is really essential mode of communication within the infected and the non-infected cells. We identify what is the unique cargo within these vesicles. Also, infected cells will produce this vesicle, but they have different cargo than the infected cells that produce vesicle. We identify small RNA that we think are priming the bystander cells, but also the unique lipids that are present in this vesicle. And one of the great advantage with vesicle that you can really target, a message in a bottle. You can really target your message or information from the infected cells to the bystander cells. And also having both enzymes and substrate in high concentration, high local concentration, can help that the signal will be more effective. Very interestingly, when we add this vesicle prior to the infection, opposite to what we speculated, this did not stop the infection, but instead made the infection much faster. So faster infection when the cells are primed first with the vesicle before adding the viruses. And so we think this is in part why we see that the blooms are almost demise in a coordinate manner. And also what we found that was very interesting, that if you mix now the virions together with the vesicle and from the infection, it will alleviate the half-time of the infection. So keeping more infectious particles in action in the water, which is very essential because UV, when you have virus or virion in the surface water, UV will very strongly decay the infectivity of the viruses. But when we mix them with vesicle, it protected the infectivity of the viruses. Interesting that I mentioned here, vesicles and communicating molecules can also help us to understand the phenotypic heterogeneity between the different states. And that's what we study now, how they can shift between the different phenotypic states during infection. And I'll show you just highlight of work of Nir Yofe in the lab, a PhD, a very talented PhD student in the lab, that was just a few months ago published. For years, we are concentrate on the demise phase that happened in the lab. This is in the lab now. Within 3-4 days, we see a complete crash. Almost 99% of the population of EHACs will die, and in parallel production of the viruses. But what happens if we wait longer? What we start to see is a full recovery of the culture, and this culture now will stay stable for many months as a resistant cell to the infection. If we now add new viruses to the culture, the population will be resistant. But it's not as simple as I just described, because when we now apply to this coexistent state, we call it, so we're shifting from lytic viral infection into coexistent, because we have not only well-growing EHACs culture, but also viral production. And indeed, when we apply now the SM fish to look how many of these cells are infected, we got this big surprise. Between 5 and 10% of the population is actually infected chronically, where the rest of the population is resistant. So really beautiful, almost symbiotic interaction that taking place here during this coexistence between the virus and the host, and with really changed the way we look now at the dynamic of the infection. Now, because we almost look at it as symbiotic interaction, we came with the following idea that if we have chronic infection produced, but the population as a whole is resistant to them, it will be a great weapon against other EHAC strains in the population, because you have a method to produce, you have a mechanism to produce infectious particles. And that's what Nir did here, using this insert, putting these two populations together, the one that we covered together with the ancestral culture. And we see very nicely the crash of the culture. There's a membrane between them, but viruses can penetrate. So we suggested that the host virus coexisted can also provide benefit to the host cells, not just for the viruses that are being produced, maybe less compared to lytic infection. So in a way, very, very interesting symbiotic interaction. And again, we took here a new single-cell phenotypic approach. Now we don't do it for RNA-seq, but Nir took the coexistent state of host and virus and sorted hundreds of single-cell, and we grew clones out of them to see, and then we re-expose them to the virus. And for our surprise, we found that the process is not binary. It's not just resistant or sensitive, but instead we see this interesting gradient of susceptibility to infections, all the way with high resistance and low production of viruses compared to more susceptible. So it's not just 0-1 condition, but within the individuality, within the population, provide this gradient of susceptibility to infection. And for us now, is really a great tool to perform comparative genomic between individuals, some that are highly resistant, some that are low resistant from the same population, from the same isogenic population, identical genetically, in order to understand for the first time new genes relate to resistance that we know basically nothing in marine algae. Antiviral defense system, this is one of our next goal. So Talia, which is another great student in the lab, PhD student now, she's using the single-cell transcriptomics in order to exactly describe now what we call the bloom anatomy and really dissect all this infection state within the same population, both in lab culture and in the field. And the fact that we have only rare subpopulation from the beginning, that although the entire population, 99%, will die in a programmed cell death manner, there's always the seed population that is resistant and recover. And using now the same approach of single-cell RNA-seq, we wish to identify this rare subpopulation that are seed for resistance in order to understand what is their transcriptomic makeup, in order to understand what is the gene responsible for antiviral defense system. And she started to generate this map. This time is the host response to the infection, and really identify very small subpopulation and see what is their gene repertoire. Very interestingly, this subpopulation we are exploring now, because they contain some of the genes relate to flagella forming, which is based back in the day, a few years ago, we showed with Miguel Frada when he was a postdoc here before moving to the Hebrew University as an independent PI, that the viral infection can cause a morphological shift from the diploid calcifying to flagellated form. So we have, in a way, internal marker now to identify this resistant cell and to understand novel genes. Another level of complexity of how we can use single-cell transcriptomic, with really a recent publication from the work led by a very talented PhD, Amir, from the lab. Now we are zooming out away from Emilia Huxley and its specific virus. And again, if we have now from the same sample we collected in the field, we have now a method to look at host-virus interaction on a single-cell level. So we can use it now to identify the still unknown host, or the authentic host, as we call it, for giant viruses. And because if we see co-expression within single cell in the field, in a way, it's single-cell metatranscriptome without even the need to isolate the virus and its host. And a major bottleneck in the field is that giant viruses discovery that was really revolutionized the entire field of aquatic virology and really changed it completely in the last 20 years. But the major bottleneck is to understand the ecological role. A lot of the work is either based on metagenomics or based on infecting of these giant viruses that can reach up to 1.5 micron and 1,500 genes, like in the Pandora virus. But most of the work was done on them when the isolation and bringing back to the lab and infecting amoeba. And that can be very unselective. And we want to use our new approaches in order to identify the authentic host of giant viruses and then to move to other viruses. And that's what Amir did. And this is in collaboration with Frank Alward, very productive and fruitful collaboration. And here, just an example between the host genes that we find in the single-cell dual transcriptomics and the different giant viruses group. And we can start to make this pairing based on prediction from bioinformatic prediction based on the data that we have from infection and dual RNA-seq.
Assaf, it's 40 minutes as you are requested.
Okay, so
Have time.
thank you. This enables us now to start to generate this map now of the co-infection within the population of different pairs, and also to identify, as we saw here, of a very unique, we have here maybe 13 individual cells to really understand also the rare virosphere and its role in the ecology, because when the bloom demise, there will be very interesting now consequence, and they can serve as an alternative host in between blooms. So that gives us now really powerful tool to identify different host virus pairs without isolation, as we did here. And also we now expanding it to different ecosystem, all the way from the North Atlantic, the South Atlantic, the Red Sea, where we study diatom blooms, and even extreme environment as alpine lakes, here in this case, in Austria, that we have now samples for single-cell transcriptomic in order to understand the dynamic and the authentic host of viruses that can really survive this extreme environment. Lastly, in the last 5 minutes, I will mention, now that we have powerful tools to identify the fraction of infected cells, we wish to understand the metabolic consequence of viral infection. So mapping the metabolic landscape of viral infection in the bloom. And I'll mention mainly amazing work of a senior postdoc in the lab, Connie Kuchlich, together with Guy Schleier, and the same mesocosm that we saw before, a different consequence, a bag that was highly infected and bloom demise, but also a bag that crashed without viral infection. And Connie and Guy applied advanced exo-metabolomics approach, which really help us to profile the unique metabolites that are produced during a viral infection. And indeed, she found a very unique set of molecules that is unique to viral-induced bloom demise. This set of molecules were uniquely, they are halogenated molecules. They're dual halogenated. They have both chloro and iodo organic compound. And for the natural product chemistry world, this is heaven of molecules that have a lot of potential, both as antiviral and antibacterial. And now Connie, in really extensive work, showed that we can see now in more than 5 different blooms that we sampled in different ecosystems, Emiliania-Huxley bloom, that we can see this unique metabolic footprint during viral-induced bloom demise. So we have very powerful metabolic biomarker that is unique to active viral infection, that is unique to the viral shunt in different ecosystems that we see the bloom. This paper is still under review, and we see this set of molecules, more than 20 molecules, not all of them present in each ecosystem, but there are some that are shared both in the endo-metabolome profile and in the exo. Very interesting, now that we have such a strong metabolic footprint from the field, and now we wish to understand what is the role of this unique molecule that we produce during viral-induced bloom demise. But that's highly challenging. And for this, Connie tried for many different ways. We have many isolates that we brought back to the lab. She tried with different infection of different EAC strain, but no molecule being produced in the lab. Only this precious moment came when she used tripartite interaction. So not only the algae and the virus, but also we add the microbiome that is associated with the bloom component. So suddenly we increase the complexity, and only when we have this tripartite unique interaction, we see production of this very strong footprint, metabolic footprint that we see in the field. So in the way, the biomarker that Connie developed here, the metabolic biomarker for the environment, is really powerful for the viral shunt, but also reporting a possible tripartite interaction that now we study in synthetic communities in the lab in order to isolate this molecule and understand its role in the interaction. So tripartite interaction, this is a major focus now in the lab in the last few years. Very interesting. One of the things we saw, for example, is that we see synergy in killing Emilia Huxley. This is when we infect the usual with virus, but when we add also the microbiome from the bloom together with the virus, we see synergy, synergistic in killing the bloom. So we call it partners in crime, and that's in a consortium. And this is when we start to have, we have a suite of dozens of isolates now from the bloom of bacteria that when we combine it with the virus, we see a much stronger and faster bloom demise that we think will be really completely new way to look at bloom demise in the ocean with really important consequence to where the carbon go and for the carbon budget. So back to the observation that we saw in the single cell fish, and single molecule fish with only 25% infected by the virus, but the entire population collapsed. This now the coalition together between the pathogenic bacteria to the algae together with viral infection can give us completely new view on what is regulating cell fate in the bloom. A few years ago, Noah Barak, that was PhD in the lab, was able to isolate pathogenic bacteria to Emilia Huxley from the bloom, especially sulfitobacter bacteria. And as we see here in culture, in co-culture, when Emilia Huxley get to stationary phase, then the bacteria can really demise the bloom in few days. Interestingly, in a new work, we show that the bacteria is really tuning to the physiological state of the host. And so they can shift in this Jekyll-Hyde type mode of interaction between symbiotic interaction to pathogenic when the algae as a dependence of the physiological state of the algae and the liquidation of the algae and the type of molecules that are being produced. At first, this is with aging of the culture, but now with the tripartite, we think that the following happened. So viral infection will produce, as we show, viral infection will lead to remodeling of the metabolism and of course lysis. So that will release a very unique metabolic molecules and signaling molecules that will induce, and this is our working hypothesis now, will induce the virulence or the pathogenicity of the bacteria, and then they can act in concert or co-infection during the bloom. So maybe the more we zoomed in to understand the viral shunt, maybe it's more like a pathogen shunt with this interaction between tripartite interaction in the bloom. And we really wish to quantify the consequence on the carbon cycle. So I want to thank so many great people in the lab that contribute to the work, our great collaborator, the funding that was generous, and for your attention. Thank you very much.
Thank you very much, Assaf, for this great talk. If there are questions, please raise your hand. There is also one question, I think, from Marco in the chat. Marco, you want to go ahead, or you want me just to read it?
As you want. Well, first of all, I wanted to say it was a great talk. Really, I really liked it. So I had a lot of questions, which I'm not gonna ask. Try to munch on your papers first. But when you were showing at the beginning of the talk this difference, the plot of the viral UMIs, where you see two populations, if I understand correctly, you were looking at a specific time post-infection, right? And you were seeing that there were different types of cells, some that were sort of infected and others were not infected, if my interpretation is correct. Okay, so I'm a physicist, so please allow me to ask stupid question here. But so it will take some time for the viruses to sort of find the host, latch onto the host, and enter into the host. So how does this dynamics actually matter on the progression of the infection that you are seeing, or can we think of it more in a sense of a very well mixed system?
Yeah, great question. I mean, I showed just one time point for the simplicity, but we have very, very detailed time point of time course of infection. What we try always to do, at least in this case, we did what's called IMOI, so adding a lot of viruses compared to hosts to saturate the system. So it won't be that we don't see infection in some of the cells because they never saw a virus. So it's saturated and mixed as a starting point. But even if it wasn't the case completely, because it's very hard to synchronize the physiology of the algae. So they are not all in the T0, they are not all in exactly, of course, same physiological state. In some cases, in other species, you can synchronize, and that for sure, I think, will reduce the heterogeneity. But we are using here the heterogeneity between the cells in order to build these small communities of subpopulation to understand the different state of infection. So in a way, it serves for us the heterogeneity in order to understand what is different between individual cells that contain the virus or they are advanced in infection compared to the ones that are not. So, but for sure, we try to maximize or at least reduce the heterogeneity at first, but we cannot synchronize physiologically the culture prior to the infection very well. But it's mixed and mixed and excess of infectious particle.
Okay, thanks.
Mark, go ahead.
Oh, absolutely fascinating talk. I've got so many questions, but I'm going to comment on one sort of strange question. This is my first introduction to these giant viruses. I can't imagine a virus that's almost a millimeter big. Now, and it's a big if viruses are transported by diffusion, the bigger the virus, the slower it's going to be transported. So the big ones might have a problem out in the ocean getting from one host to another. Any possibility that they're latching onto the bacteria, which are modal, so that you get both the virus and the bacteria going at the same time?
Sounds everything is possible for sure. There are also, what helps also in viscosity is the fact that they produce this polysaccharide, these sticky polysaccharides during infection. So that can be also, and we see that can be also a kind of a trap for the viruses, that even when we see sinking particles, that can be, from one hand, we are thinking of it as a possible resistant mechanism because you can mask by this polysaccharide, you can mask the receptor to the virus. And from the other end, you are stuck with your viruses, you are trapped to it. And that maybe help to local infection. Because one of the interesting questions, these massive blooms are produced maybe once a year annually. But then viruses need to survive over 8 months, with UV and so on, that is damaging the viruses. So there must be some kind of mechanism to keep the reservoir of the viruses for the next bloom that will take place. So it's an interesting question, how do you keep a reservoir of these viruses and diffusion within a subpopulation one way maybe to do it is I show that they are not only lytic, they are not only killing the cells. Some of them are budding out of the cells, what is called chronic infection, as I show in the coexistent. So maybe this is, a small reservoir that is accumulating in individual cells as a seed for the next year bloom, and a mechanism also to move by diffusion locally between the individual cells.
Thanks. Amazing work. I'm really just fascinated by it.
Thank you.
Any other questions? I have one, actually, maybe two. So you showed that there are these extracellular vesicles. Right. This is very intriguing to me. So Emiliana Haskell is continuously producing those cells regardless of the infection state?
Yes.
And then these vesicles might help the virus to sort of camouflage, or?
Yeah, so every cell will produce vesicles. Every cell in the ocean, every cell in our body will produce extracellular vesicles that are called exosomes. And every organism will produce vesicles. But the question is, what is the unique cargo that you have in different states? So in a way, the viruses are hijacking this mechanism of extracellular vesicle production. And when they infect, we see that the cargo is very different from vesicles that are produced in control uninfected cells. And the first work, actually, an extracellular vesicle came from Penny Chisholm lab in cyanobacteria, in Prof. And back then, they hypothesized that actually vesicles are a kind of defense mechanism against the infection, because the phages can inject their DNA into the vesicle. So it's acting like a mock infection, as a trap, and not infecting the cell. But this was never proven in cyanobacteria, but it's definitely widespread in the ocean. And the numbers, although viruses are considered as the most abundant entity in the ocean, with up to 10 million particles per ml, vesicles are, can be very important fraction of this extracellular milieu.
That's great. One little more also, which is maybe outside of the scope. Because of these tight interactions between the virus and maybe even the giant viruses and bacteria also, have you by any chance checked for horizontal gene transfer?
Yeah, so very interesting. At least between the host and the virus, we definitely see pathways that are shared. And I mentioned one pathway, which is for unique set of lipids that are called sphingolipids. They are highly homologous to the host. But, in the world of phages and bacteria, usually the idea of horizontal gene transfer that the phages collected or grasped genes from the host. But in this case of giant viruses, there are a lot of controversial in the evolutionary world that maybe these giant viruses were in evolution came even before the first eukaryotic cell that was based on RNA, because these giant viruses are always double-strand DNA. And so in a way, gene transfer can go also from the virus to the evolution of the host, of the eukaryotic host. And definitely gene transfer for several metabolic pathways, very common in the sea.
Great, thank you very much. Any other questions? We're at the hour. Okay. Well, thank you so much again, Assaf. That was a fantastic talk, and it's a great work. Thank you, everyone, for joining us, and we'll see you next talk next month. Bye.
Thank you very much. Bye.